Thank you, Professor Chai. Good evening, everyone from Bristol, UK. Today I'll be presenting the effects of modulating cyclic GMP and protein kinase G, PKG, on nerve-mediated neurotransmitter release from efferent nerve terminals in the mouse detrusor. ATP is a co-transmitter with acetylcholine and parasympathetic nerve terminals, and it has an excitatory role in generating nerve-mediated contractions in the detrusor for most animal bladders. ATP is released over lower stimulation frequencies, whilst acetylcholine release predominates at higher frequencies. However, in the human bladder, ATP only has a role in bladder pathologies such as overactive bladder. Therefore, it would be highly desirable to selectively reduce ATP release from nerve terminals and leave acetylcholine release intact. Phosphodiesterase type 5, PD5 inhibitors, like sildenafil, inhibit cyclic GMP breakdown, and in previous studies we have shown that it reduces nerve-mediated contractions and specifically the lower frequency purinergic component of nerve-mediated contractions. We have also demonstrated that sildenafil reduced the amount of nerve-mediated ATP release measured. In this study, we hypothesized that increasing intracellular cyclic GMP levels by different pathways decreases ATP release. The effect on acetylcholine release is also not known, and this was tested in the study. Intact mouse bladder tissue strips were used for conventional tension recording experiments, and contractions were generated by electrical field stimulation. Tension was plotted as a function of stimulation frequency and analyzed to estimate parameters like maximum tension, Tmax, and the frequency to generate half maximal tension, F half. A reduction of Tmax implies an action on force mainly by acetylcholine-dependent pathways, and an increase in F half is indicative of a reduction of force at lower frequencies, whilst leaving force at higher frequencies unaffected, and therefore it implies a reduction in force mainly via ATP-dependent pathways. Nerve-mediated ATP and acetylcholine release was also measured from superfusate samples. So over here, you can see that sildenafil has resulted in a reduction in nerve-mediated contractions with a greater effect at lower frequencies. This is reflected in the force-frequency relationship parameters, where you can see that sildenafil has resulted in an increase in the F half, and this corresponds with a reduction in ATP release. There was also no effect on maximum tension, and no effect in the amount of acetylcholine release measured. Sildenafil increases intracellular cyclic GMP by reducing its breakdown by PD5 inhibition. Another way to increase cyclic GMP levels is by increasing its production by enhancing guanylate cyclase activity. Using a soluble guanylate cyclase activator, like sildenafil in this instance, also reduced the amount of nerve-mediated ATP release measured, and subsequently reduced nerve-mediated contractions. There was an increase in the F half, and there was no effect on the maximum tension, demonstrating a greater effect at the lower frequency purinergic contractions. Sildenafil also had no effect on acetylcholine release. Using a cyclic GMP analog, 8-bromocyclic GMP, to directly add to the cyclic GMP intracellular pool also had a similar effect, reducing nerve-mediated ATP release and nerve-mediated contractions. The intracellular target for cyclic GMP is protein kinase G, PKG, and using this PKG activator had a similar effect on the force-frequency relationship profile, where there was an increase in the F half and there was a lack of effect on maximum tension. Using a PKG inhibitor had no effect on nerve-mediated contractions and ATP release by itself. However, it did inhibit the effects of when we increase levels of cyclic GMP with sildenafil, so that now sildenafil, in the presence of the PKG inhibitor, had no decrease in nerve-mediated ATP release, and there was no increase in the F half value when looking at nerve-mediated contractions. This suggests that the intracellular target for cyclic GMP in regulating neurotransmitter release, well, at least in regulating neuronal ATP release, is PKG. In this study, we have demonstrated selective inhibition of neurotransmitter, namely ATP release from nerves supplying detrusor smooth muscle, elevation of intracellular cyclic GMP by several different routes, whether it's enhancing guanylate cyclase activity with sinusigot, reducing breakdown of cyclic GMP by PDE5 inhibitors like sildenafil, or directly adding to the intracellular pool by using a cyclic GMP analog PKG activator, all selectively reduces neurotransmitter release to detrusor smooth muscle. The intracellular target for cyclic GMP in regulating neuronal ATP release is PKG. We see an attenuation of ATP release whilst leaving acetylcholine release intact. Because ATP is associated with pathological contractions in the human bladder, it offers a therapeutic target to selectively attenuate these contractions whilst leaving physiological contractile regulation intact. So I'd like to thank everyone who has been involved in this project, funding bodies, the ICS for giving me the opportunity to present on this platform, and thank you for your kind attention.